I'm going to hand the mic over to Russell Vance, who's a faculty member in the Immunology and Pathogenesis Division of MCB, who was the chair of this. He was the chair of the meeting.
Great, thanks, Dan. So it's really my delight to be able to introduce to you Elizabeth Winzeler, who's getting her computer set up now. Elizabeth got her start, I guess it's sort of maybe a theme for today, got her start doing some pretty basic microbiology studies in Caulobacter in Lucy Shapiro's lab. That's what she did her graduate work at Stanford. And then for her postdoctoral work, she moved to Ron Davis's lab, also at Stanford, where she really applied, was one of the first to really apply some of these genome-wide technologies in yeast to really address, biological questions. And then she did something fairly brave and inspiring, I think, which was that she jumped from yeast to a completely different organism, malaria, when she established her own laboratory, first at the Genomics Institute of the Novartis Research Foundation and at Scripps Research Institute. And she's been working on malaria, again, applying genomic-wide technologies to try to understand the biology of this very interesting and very complex protozoan. So, and I think she's really done some very interesting work. So thanks very much, and I'm delighted that you're here.
Okay, thanks. I've titled my talk non-traditional, and what I mean by non-traditional, I'm going to talk a little bit about virtual chemical and brute force or industrial genetics of malaria. And hopefully all of this will become apparent what I mean by it as we go through the talk. So malaria is caused by 4 species of protozoan parasites of the genus Apicomplexa. It's transmitted by the bite of female Anopheline mosquitoes. And malaria is a major economic burden on countries where it's endemic. And I've shown 2 maps here from the World Health Organization website, one of which shows the estimate of world poverty, and here is the estimate of the world malaria burden. And as you can see, they overlap pretty well. And there are some people that would say, well, you have poverty and then you get malaria, but there are others, such as Jeffrey Sachs, who have argued that you have malaria and then you get poverty. And this is based on the fact that if you're in a malaria endemic region, many of the workers are staying home from work because they are taking care of sick children. It's one of the most significant causes of absenteeism in Africa, and it's estimated to decrease African growth by about 1.5% per year. There's currently no licensed malaria vaccine, even though you've probably heard some news about RTSS and the subunit vaccine that was developed by GlaxoSmithKline. This is headed for licensure in about 2011, even though it is an imperfect vaccine and would probably only give about a 50% reduction in the number of malaria cases and probably would not be licensed in the United States. Currently there's about 250 to 500 million cases. It looks like we have a downward estimate, and I'll go into that a little bit later. And most of the people that are affected are children under 5. So this is just a picture of what the disease looks like. This is a woman taking care of her sick daughter. And there are about 2 to 3 million children will die usually each year. And what happens, falciparum malaria can produce relatively severe symptoms. Vivax malaria, which is found in most of the rest of the world, is not quite as severe. But this is what happens in falciparum malaria, where the parasites develop knobs on the surface of the and the infected erythrocytes end up adhering to your brain, and this results in an unarousable coma, swelling of the brain, and eventually, you can end up dying. So I am actually mostly interested in drugs. So I just want to tell you why malaria is such a big problem these days. And part of it is not that you can't get rid of malaria with drugs, it's because resistance is a really big problem. And it partly has to do with the way that anti-malarial drugs are used in the world. They're often given, they can be purchased from the grocery store versus going to a doctor. People usually take them until they feel better. They are distributed in places like Western Thailand and at the Cambodian border, where there's a lot of counterfeiting going on. So in many cases, the drugs will be diluted or ineffective. And so we've seen resistance emerge very rapidly after each new drug is introduced. So this is the traditional cure, quinine. It is found in gin and tonics, which was, as many people know, why the British were drinking gin and tonics in India when they were occupying it. It was because it has an effect against malaria. Chloroquine is a synthetic version of quinine that was introduced in 1945, very successful drug. Took many years, actually, about 11 years before you actually observed resistance. And this has probably been one of the most successful drugs so far. The other ones, resistance developed very rapidly in the field after they were introduced. Typically, we target the the folate biosynthesis pathway, or you have aminoquinoline such as mefloquine or atovaquone, which inhibits mitochondrial function. So in all cases here, we have resistance to these, and the only thing that's out there, this is our last line of defense currently, is an artemisinin combination therapy. It's based on a natural product from the wormwood plant. This is it shown here, I think you guys all know about this one because of Jay Kiesling. But anyway, this has been relatively difficult to synthesize. It was introduced in 1996. There's currently no verified cases of resistance, but disturbingly, there have been, people have noted that there's an increase in the time to parasite clearance if you get parasites from Western Thailand. So people have been using the natural product that gives rise to coartem for many years in this region. And it now takes probably about twice as long to actually clear the parasites if you take artemisinin-based combination therapies than it took about 10 years ago. Although the resistant parasites have now been introduced into the laboratory, and it's really been very difficult to pin it down. So it looks like it's a little bit more complex than a single point mutation. Anyway, so we would love to have a malaria vaccine. It's proved difficult. People have worked on it for a number of years. So people have put a lot of research and development into anti-malarials. And as I'll show you, it's not actually that difficult to create anti-malarial drugs, but what we need to do for treatment for drugs that are going to work in Africa is create anti-malarial drugs that are inexpensive, cost pennies per dose ideally, that are completely stable at room temperature, that have low toxicity, that are preferably hitting a novel target, so there's not going to be so much probability of cross-resistance. And we also need to just keep things in the pipeline because we know that resistance will likely emerge, even if we try to distribute them using combination therapies. So there are three major challenges in malaria research, according to me. One is to develop new drugs against novel targets, and again, these need to be inexpensive, long-acting, well-tolerated, and very easy to administer. We need to develop a useful and inexpensive malaria vaccine, preferably one that is able to actually prevent malaria. And we also need to increase our understanding of the molecular basis of malaria in order to facilitate drug and vaccine discovery. And as someone who does a little bit of both, it's very frustrating to me how difficult it is to actually do things in malaria because there isn't a lot of knowledge about parasitic processes, that we don't have the tools that everyone takes for granted if you work on a model system. And there's very little knowledge, very few students that have actually been trained to work in malaria, and so you basically have to reinvent everyone from scratch. And there's also a very low density of researchers working on malaria, and this makes it difficult to actually create a community that can facilitate drug and vaccine development. So the first part of my talk is going to be using virtual genetics to characterize the parasite life cycle. And I was trained in classic genetics, classic forward genetics. I was in developmental biology. And you know, what we always used to do in developmental biology at Stanford was to do forward genetics. And many of you are familiar with this type of Drosophila that has two sets of wings here. And the classic way for doing forward genetics is you identify mutants that can't grow, and you can then characterize them based on a set of phenotypes that they share. You can create complementation groups based on the similarity of your phenotypes. You can map phenotypes to different genes, and this allows you to delineate regulons that are of sets of genes that are all involved in a particular process. Well, you can't really do that very effectively in malaria parasites. For one thing, they grow primarily in liquid culture. So you can't really get nice clones. And you can't really mate falciparum malaria parasites because you actually need a chimpanzee host in order to do that. So this is the life cycle shown here. Chimpanzee or human, I should say. This is the life cycle here. So it's transmitted by the bite of an Anopheline mosquito, which releases thousands of sporozoites into your bloodstream. These migrate to your liver. If you have vivax malaria, it sets up a hypnozoite, but eventually they develop in your liver. And this one sporozoite will give rise to about 10 to 20,000 merozoites, which are then released into your bloodstream. And they synchronously, semi-synchronously can infect your different red cells, set up an infection. This cycle is accompanied by chills and fever. You develop anemia, and in some cases, if they end up in your brain and cut off blood flow to the brain, you can end up dying. We don't understand really how this life cycle is actually driven, or at least we didn't when I started working on it. In response to some cue that we don't understand, they form male and female gametocytes. We don't know actually the basis of sexual development in malaria parasites or the basis of the sex chromosome. They are taken up by the mosquito, and they undergo a whole developmental cycle within the midgut of the mosquito. They're haploid throughout this stage here. They mate with one another. Within the mosquito midgut, you have a brief meiosis. They migrate across the midgut, form an oocyst, and eventually they migrate to the salivary glands. So there's a huge number of things that aren't known about this whole cycle here. For example, I think it's just going to come out, but at the time we didn't know the identity of a single transcription factor that controls this very complicated developmental pathway. So there's about 6,000 genes here. We know that there are genes that are coming on and going off during all these different cycles, throughout the cycle, but we didn't really know how any of this is controlled. And much of the malaria research over the past 30 or 40 years has simply been devoted to creating compounds and putting it on these parasites instead of trying to understand how the parasite is actually put together. And it is relatively difficult to work on. So I'm going to talk about how we're using virtual genetics to try to characterize this process. So we know from work in our lab that genes involved in similar processes or pathways tend to be co-transcribed. And what we've been doing over the past 7 or 8 years is collecting very detailed gene expression data for as many life cycle stages and conditions as possible, with the idea that highly correlated expression patterns over many different conditions may form regulons. And this is just a subset of some of the data that's come from our lab, where we've taken RNA from the erythrocytic cycle, we've taken RNA out of sporozoites, we've taken RNA from liver stages, done subtraction on it, and then we put all of this data together to create groups of genes, to predict regulons. And it's, again, it's difficult to work on malaria, but it's relatively easy to obtain RNA from many of these different life cycle stages. Again, sporozoites, oocyst. You can induce gametocytogenesis. You can do subtractions on liver stage material. And all of this, again, goes onto an Affymetrix chip that we built a while ago. And you can put all the data together, and you create these red-green color maps. And then we'll use bioinformatic tools to try to create genes that we think, sets of genes that we think are going to be involved in a process. And I'm not going to go into all the tedious bioinformatics, but we have various parameter optimization routines that we use to set the boundaries of these different clusters. And we take a group of genes that we think is involved in a particular process. For example, someone may go to the literature and someone working in the field for a number of years may have identified a group of 5 or 10 genes that they think is involved in the process of invasion. So this is the process by which a parasite, latches onto the red cell and then goes inside. And so you can take the genes that are known to be involved in cell invasion, and then you can try to create, minimize different parameters so that you can capture the largest number of cell invasion genes in an expression cluster as possible. And then when you do this, what happens is that you've got a group of about 35 or 40 genes that contains about 8 of the 9 known cell invasion genes. And the probability that you would get 8 of the 9 cell invasion genes in a group of 6,000 by chance is about 1 times 10 to the minus 17th. And then you end up with a whole bunch of hypothetical genes that are very likely to be involved in the invasion process that just came along for the ride. And so you can do all of this without actually going into the laboratory and trying to do forward genetics. And this actually works quite well. And there's various bioinformatic proofs. For example, this is I'm not really interested in the nucleolus, which is an organelle involved in RNA production. But these are the plasmodium genes here. These are the yeast genes here. And this was just a cluster of genes that was created through expression profiling. And it turns out that many of these genes were just listed as uncharacterized, as are 50% of the genes in the malaria genome. And in fact, if you go and you look at their yeast homologs, you find in most cases they are known to be involved in nucleolus function. So it really actually works extremely well. And this has given us a large number of different candidate genes that can be used for experiments down the road. And this is not just, I mean, a lot of people say, well, you work on malaria, it's not really relevant to anything that I do. But it actually can serve as a model organism for humans. So what we do here is that we've created an expression cluster. This is, again, now going into a process where most of the genes are listed as hypothetical. And these are the genes that are upregulated as the parasites exit the asexual stage and begin sexual development. So they turn into the male and female gametocytes. And this is the cluster of genes that's upregulated when the parasites turn into male and female gametocytes. And here are some of them. And you can take actually the homologs of some of these genes, and you can actually look at how their human homologs cluster. And we've taken data from a large number of tissue samples that were collected by my colleagues at Novartis at the Genomics Institute in San Diego. This has been clustered, and it turns out that if you go back and forth between the two, the genes that cluster with sexual development and malaria parasites will also cluster, are also upregulated in testes in humans. And it seems that what both the male gametes have in common in malaria and in humans is that they both have a flagellum, and probably the genes that are involved in flagellar function are conserved throughout evolution. And of course, you can bring in lots of other high throughput data sets. It's relatively difficult to work on these things one by one, and you can validate many of your predictions. And ultimately, what we're doing is beginning to use sort of virtual genetic strategies to define regulatory sequences. So you can then take all of the genes that are upregulated under a particular condition. You can extract the regions upstream, and then you can begin to deduce what sequences are controlling development. And again, various bioinformatics strategies that we're using, which you can read about if you're interested. And after you go through all of this, you can find sets of motifs that are likely to be involved with different processes as you move through developmental cycle. And these were just some of the ones that have come out. And then we can go even further, and you can begin to do alignments and gene disruptions on the motifs. The ROP tree is a specialized organelle in the malaria parasite. Parasites bind to the red cell. They essentially the ROP trees are sort of like oil can type structures at the end of the parasite, and they hold a bunch of proteins. And when the parasite binds to the red cell, it basically squeezes all these proteins into the red cell membrane, and then it sort of sucks itself inside. And so most of the proteins that are known to be involved in ROP tree function all have this motif shown here. And you can actually begin to take hypothetical genes that contain the motif, and you can look at related apicomplex and organisms, and you can find that they actually are found in the ROP trees. So it also becomes a way to predict new ROP tree genes. So we've identified a large number of motifs, and the question then is, what are these different motifs doing for the parasite? And again, we're using kind of a reverse genetic strategy. These are the motifs. We've identified a new family of Manuel Linus at Princeton identified a new family of DNA binding proteins called AP2 proteins, and he used a protein binding microarray to predict the sequence of these recombinantly expressed proteins. And lo and behold, several of these sequences that were bound in a protein binding microarray appear to be identical to the ones that we were able to identify by gene expression profiling, which now allows us to sort of use systems biology to begin to put transcription factors together with the sequences that they actually regulate. So that's just virtual genetics, and hopefully I've showed you that in malaria we can identify regulons by gene expression profiling, and again, data produces probabilistic models for the functions of parasite genes, and we can find likely transcription factor binding motifs, and that this can be used to identify possible targets for drugs and vaccines. So I'm going to switch now to chemical genetics of malaria, because I have some associations with the Genomics Institute of the Novartis Research Foundation, which was an institute that was set up by Pete Schultz about 10 years ago. And we do lots of high throughput stuff there. So if you follow the drug development literature over the past 10 years, you know that people talk a lot about target-based screening. So here you find a biochemical target that you think is interesting, maybe it's a DNA polymerase or something. You set up a biochemical assay, and then what you do is that you then look for sets of compounds that are able to inhibit that process in a microtiter plate. And the problem with this target-based screening, which was very popular in the 90s, is that in many cases you end up finding compounds that inhibit your biochemical process, but which are unable to clear an infection, presumably because they cannot get into a cell. Furthermore, even though you can validate your target using all sorts of beautiful genetics, in many cases it's really difficult to absolutely predict which targets are truly essential for the parasite, and which when inactivated will give you rapid parasite death. It may also be difficult to develop assays or to overexpress a given protein target. It can take a person a year or two to actually get any particular target expressed. So we've been using cell-based screening, in which you essentially screen all targets simultaneously. And the idea of this is that we can rapidly find the compounds that are going to work, and not only this, but it may produce chemical tools that can be used to address the function of genes. So many of these compounds that I'm going to describe will not be actually making any drug, but they may eventually give us tools that we can use to investigate malaria parasite biology. So we developed a 1536-well assay a few years ago in order to identify compounds which inhibit malaria growth. And we were able to run this. This is our robot shown here. And again, I have to, of course, acknowledge all of the great technology that was set up at GNF by Pete and his friends. And we were able to use the robots. All of the compounds are stored in semi-desiccated. Basically, it's all completely computerized. So you don't actually have to worry about what's in any of these wells for the most part. And the compounds, there's just a huge sort of, I guess this is relatively common in all the drug companies now, but it was kind of cool about 10 years ago. But anyway, we have 2 million compounds that are the combination of the Novartis and the GNF libraries, and we were able to develop an assay that worked in essentially 1536 well plate. And basically what we're doing is essentially reading you take your compounds, you put them in a microtiter plate, you add your malaria parasites, you let them sit there for 48 hours in a low oxygen environment, and then you read the plates looking at the increase in DNA content about 2 days later. And this is just showing the DNA content in the parasites. They grow in red blood cells that don't have DNA, so this is relatively easy. And through running this assay, we were able to identify about 8,000 compounds that were able to essentially prevent malaria parasite replication with EC50s, most of them of less than 1 micromolar, and at least 600 of them had EC50s of less than 100 nanomolars. And we were able to identify many novel scaffolds with no apparent toxicity, and we were able to do sort of PubChem searches that showed that we were able to basically rediscover most of the known antimalarials with less than 1 micromolar activity. So the advantage, so I'm really interested in systems biology, and so we're beginning to apply the same sorts of tools that we've used to analyze our gene expression data to analyze the chemical screening data that we've been collecting. And so you can use Mike Eisen's cluster program, which I showed you before, to cluster now the chemical screening data that we collected. So before we were looking at gene expression data as you went through the life cycle, and now what we can use is we can collect all of the historical screening data that have been run on all of the compounds that was in this collection. So over the past 5 or 10 years, we've run this set of compounds against at least 200 different assays that were all done at GNF. And all of that data has been put into our Cheminformatics database. And you can then take all of this historical screening data, and you can then cluster, and then take your anti-malarial hits, which are shown here, and then you can cluster it, which allows you to rapidly identify those compounds which are likely to be broadly toxic or to come up in every different screen that you run. So for example, this would give you something like tenopal or is it blastocyte? No. It's that pore-forming antibiotic. But anyway, these things would be these are not things that you would want to turn into a drug because they would probably kill you just as well as they would kill the malaria parasite. And then what we're really interested in are these things up here that are going to be very specific for malaria parasites, but are not going to come up in any of the different human assays that have been run. And again, we can actually validate the screen, again, using Cheminformatics. You can take each one of these chemical entities. About 5% of them actually have annotations, and you can do PubChem searches for them. And one of the things that was kind of cool about this is that it allowed you to actually determine the mechanism of action of some of these compounds. So the basic problem with cell-based screening is that you can get lots of hits that work really well, but you don't know what they're doing. And people will say, well, you know, it's going to be so many years down the road before you actually find out what this compound is actually doing in the cell. So what you want to be able to do is begin to try to use the Cheminformatics to begin to characterize what the compounds are likely to be doing. And so this is just one of the clusters that came up from our analysis here. And these are about 200 different screens that were run, and the red and the green shows whether it was active or inactive. And for this group of compounds here, they were all active against malaria parasites, which is why you have a little red line here. But they were also tended to be active against some cell lines like BCR-ABL cell lines, cancer cell lines. And it turns out if you take this group of compounds that all tended to end up together based on hierarchical clustering, it turned out that almost all of them actually had the same pharmacophore. And PubMed searching would actually identify that almost all of them are hitting the folate pathway. So I'm not sure you can see it, but they all have a diaminopyrimidine, and you have many of the known antifolates, such as pyrimethamine and various other types of genes, compounds that hit dihydrofolate reductase. And so we haven't used any of the chemical structure information into creating this cluster. It was solely based on their activity against a large number of cellular screens. And actually, what's interesting is that you actually have structure coming out of that exercise. And the probability that you would have all of the 15 known antifolates within this cluster by chance can be estimated at 1 × 10 to the minus 38. So it's actually quite remarkable. So this is not just an academic exercise. I'm involved in a drug discovery consortium that involves the Novartis Institute for Tropical Diseases, which is the drug discovery end of what Rina Rupali had described earlier. And the goal is to work with public funding agencies, including the Wellcome Trust and the Medicines for Malaria venture, to try to bring some of these lead compounds that we identified in this screen to turn it into a drug. And I'm going to be talking about the work of lots of different people that are part of this consortium, but the goal is to create a one-dose cure for Plasmodium falciparum. And this is just pharma speak here, clinical selection point candidate by the end of 2009, and then to create something that will be ready to hopefully go into man by the end of 2011. And this is just some of the people that are working on this. And we had originally done this assay on a set of natural products that came from the Novartis Natural Product Library. And we started this in about September of 2006. And this was chemistry mostly being done at NITD with the hits that came from the screen that I just described. And we're now at this point right here where we've shown that some of these compounds are able to completely cure mice. And of course, this is lots of work. And I am not a pharmacologist, but this was the original lead compound here. And through optimizing the different scaffolds, we're able to create a compound. The chemists at NITD were able to create a compound that have all sorts of desirable characteristics in terms of exposure, protein binding, solubility, and so forth. So at this point, we were able to put this compound into mice. And at this point, giving 3 doses at 3 times 30 mg per kg over 3 days, we were able to achieve a complete cure of all of the mice that were put through this, a 99.9% reduction in parasitemia. And this compound is actually comparing very well to the standard drugs, including artesunate, chloroquine, and methylquine, none of which are able to give a complete cure at 3 times 30 mg per kg. So the last few minutes here, I'm just going to talk about what I call brute force or industrial genetics. And this relates to the question of how do you determine what many of these uncharacterized compounds are likely to be doing in parasites. So I'm a big fan of microarrays. And so one of the things that we've done over the past couple of years is to basically make a tiling microarray that goes through the genome at basically two base spacing. So malaria parasites are haploid, so we don't have to worry about some of the complications of having a diploid genome. Essentially, you can take genomic DNA from the parasites, you can chop it up, and then you can basically detect every single base pair change by comparing the hybridization patterns of your reference strain to your drug-resistant strain here. And this is great for looking at how compounds are actually acting, or how resistance emerges at a single nucleotide resolution. But one of the things we're also doing is using this to sort of do population genetics and study how parasite strains move through the populations in Peru or in Papua New Guinea after drug selection has been applied. And this is just an example. This is the chloroquine resistance transporter. We're not doing any amplification of any of this. You just take straight genomic DNA, you digest it, you put it on the chip, and you run it through some fairly complicated software, and you can detect most of the known point mutations in the chloroquine resistance transporter, a gene which has been studied intensively for many, many years. And one of the things that we've seen very frequently, very early on, is that a lot of the drug resistance actually turns out to be copy number variation. This was identifying the target of fosmidomycin. This is a potential anti-malarial. It's fairly potent. And my collaborators at Columbia University, David Fiddock, was able to evolve fosmidomycin resistance, and it was thought that the target was a deoxyxylose phosphoreductoisomerase. And he went through and sequenced it, and he could never find any point mutations in this particular target. And so what he was able to do was he gave it to us. We put it on the chip, and just in a single overnight hybridization, it comes back, and we put it on. We looked at it, and it turns out that it has an amplification on chromosome 14, and the first gene in the amplification is actually the predicted target. And we've seen this in a number of cases here, that we see gene amplification events that seem to be associated with the gene that's actually the target of a drug. So I hope I've shown that cell-based screening is an attractive alternative to target-based drug discovery, and that advances in cheminformatics, genomics, and high-content imaging may make the daunting task of determining what compounds are likely to be doing in the cell less daunting, and that this approach has actually led to compounds that are able to clear experimental and malaria infections with a single, well, actually 3, I think it is a single dose if you use a higher amount. So, and so I've had many different collaborators at GNF and also through my Scripps lab. Most of the academic work was the work of Nikesh Daria and Jason Young, two a current and a former student. And I've had a number of different collaborators throughout the world who have given me samples and helped my entry into the malaria world, and support from the various different foundations.
Okay, so if there's, we'll have a few questions and then lunch after that. So, there's going to be a panel during lunch, but yeah. So first we'll have any questions. I was wondering what if you figured out what the target of your new drug was.
No, not
and
yet.
how easy it is to evolve resistance to it.
We're working on that right now. We're also doing affinity chromatography. It takes like a year to get enough pellet to actually run an affinity chromatography experiment. It's kind of unbelievable, but My hypothesis is that it's inhibiting protein biosynthesis. But
did the gene cluster that you found when they came out of asexual phase, do those genes cluster anywhere on a chromosome? Can you start to identify the...
Not much more than what would be, slightly higher number than what would be expected by chance. But it's similar to, for example, Saccharomyces, where in many cases you would have two genes that are divergently transcribed, which show co-transcription within a life cycle, but are probably adjacent to one another on the chromosome because they're co-regulated. So, but not generally.
Making me run. Okay.
The drug assays that you were telling about, the 48-hour assays, like it's just you are settling the cells on the plate and you are measuring the DNA content after 48 hours. So is the a lot of factors change, like the media is being used up, the red blood cells are being killed. So are these being replenished or are they being kept static for 48 hours?
It's being kept static. So you do the infection at a very low parasitemia, so about 0.5%. So after 48 hours, you typically only go up to about 1 or 2% parasitemia. And then there's always a very big excess of uninfected red cells. So yeah, that's all taken into account in the assay.
Okay, so if there's no more questions, let's thank Elizabeth.